In lecture 1, we talked about the different agents of evolution. We talked about mechanisms that could cause populations to evolve. But my question is, why on earth did we even think populations evolved in the first place? Why do we think that there was a common ancestor to all living things? So in this lecture, we're going to take a look at the evidence that made us believe that populations were evolving over time and that there was a common ancestor to all living things. Now this information in this part of our notes is just simply to show you that we've collected data and information and we've made observations from many different branches of science that support the idea that populations and species have evolved over time. So for example, molecular biologists, they've observed that there's many common molecules and processes found in many different organisms. Those that study anatomy, we find that there's anatomical structures that are very similar in different species. Why is that the case? People that study embryos, embryology, they've noticed that there's many similarities in the development of embryos in all species. Paleontologists, they provide us fossils. So we actually have remains of organisms that existed or that were ancestors to some of the modern populations and species that we have today. And then finally, we can use things like mathematics to justify when organisms were here and what would be possible ancestors to some of the species that we have or some of the species that we find that are fossils. So let's take a look at specifically what is the evidence out there that supports the idea that species have evolved over time. Well, the first group or broad category of evidence is what we call molecular homologies. And you know homo means the same. So when you see homologies, you can just substitute in the word similarities. There's molecular similarities between different species, and this indicates a common ancestor. Or we can use this information to actually determine how related two species are, or how distantly related two species are. Molecular homologies, or biochemical just basically means molecules. Molecular homologies are similarities in the molecules or cellular processes of different species. So let's take a look at some examples of molecular homologies. We've already discussed many of these before, so I hope this is a review. We know that all organisms use the same 4 bases in their genetic code, A, T, C, and Gs. We find that making up the DNA of all organisms. We know that organisms utilize DNA and RNA. All organisms utilize ATP for cellular energy. We know organisms use the exact same 20 amino acids to make their proteins. The codons that code for these amino acids are the same for all organisms. And we see the wobble effect in all organisms. We see the same wobble. So for example, CUU, that codon codes for leucine. And we know that third letter, that third base can wobble to a different base and it still codes for leucine. This occurs in plants. This occurs in fungi. We see the same wobble in animals. So the fact that we see this similarity in all species indicates a common ancestor. We can also take a look at proteins. Again, we're talking about molecular homologies, and proteins are molecules. So we can look at similarities in proteins in different species. One protein that you should probably have memorized that all species on our planet, from bacteria to fungi to plants to animals, all of us possess this, is cytochrome C. Cytochrome C is a protein that's found in the electron transport chain of all organisms, and it's necessary for the production of ATP. Another protein that we can find in all organisms to analyze is ATP synthase to produce the ATP that organisms need. Hemoglobin is another one that we commonly use, but I want to point out we can't use hemoglobin to determine how closely related we are to fungi because not all species have hemoglobin for us to analyze. But if we want to see how closely related we are to yeast, which is a fungi, then we can take a look at our cytochrome C proteins, and we can take a look at yeast cytochrome C proteins, and we can compare how similar they are. And that's what these data tables are attempting to show you down here. They're showing you how similar different proteins are between other species and humans. So this first one is cytochrome C. And if we take a look at cytochrome C proteins of chimpanzees, and we compare them to our cytochrome C proteins, there is absolutely no difference. Their amino acid sequence is identical to our amino acid sequence. If you can compare us to a rhesus monkey, they have one amino acid that's different in their cytochrome C. So the fact that we have no differences between us and chimps just indicates a very recent common ancestor. We are more related to a chimpanzee than we are to a rhesus monkey. And as we go further down the chart here, it makes sense that we are least related, according to this chart, to yeast cells. Yeast are a fungi. And we have some similarities, but we have many differences between us and yeast. And we can see that in our cytochrome C that they have 42 different amino acids compared to our amino acid sequence. And so that just indicates that we're more distantly related to yeast than we are to pigeons or cows or rabbits. Over here, it's showing you the difference in structure of hemoglobin, which again is a protein that is in your red blood cells. And we can see that if you take a look at hemoglobin proteins in red blood cells of gorillas versus ours, there's only one difference. There's only one amino acid difference. So we are more related to gorillas than we are to rhesus monkeys, according to analyzing our hemoglobin proteins. Another similarity that we see is that all organisms contain ribosomes. Prokaryotes have ribosomes as well. They're smaller than eukaryotic cell ribosomes. So again, I want to emphasize, we can call these similarities molecular similarities or molecular homologies, or we can call them biochemical homologies. Biochemical is just referring to chemicals, referring to molecules. But we can also see similar processes that occur in different organisms. So here's some examples of some processes that indicate a common ancestor. The fact that we see the flow of information from DNA to RNA to proteins, which is referred to as that central dogma, indicates a common ancestor because we see this flow of information in all organisms. We also know that all organisms perform glycolysis. Even bacteria perform glycolysis to generate ATP. So that is a process that we find in almost all organisms, which indicates a common ancestor. So not only should you be able to list me some of these molecular homologies that we just discussed, but you have to be able to use data related to molecular homologies to draw conclusions. That's what they're going to ask you to do on the AP exam. So here's what I mean by that. I've given you data related to a molecular homology. It says determine which species shares a most recent common ancestor to humans. This is just a fancy way to say who's more related to us, according to this data. So the data they're giving us, again, is just the difference in the amino acid sequences of hemoglobin proteins found in the different species. And because gorilla hemoglobin proteins are very similar to ours, there's only one amino acid difference, then our answer would be the gorilla. And we would be able to justify that because we would talk about how similar their hemoglobin is compared to ours, and that indicates a common ancestor. The next question says, draw a phylogenetic tree to indicate the most likely evolutionary relationship between humans, gorillas, and rhesus monkeys. So again, we're using this data about this molecular homology. Now we're going to do more with phylogenetic trees. I'm just going to show you a basic one here. If we say that rhesus monkeys are less related to humans, then we're going to show that there was a common ancestor between us and them a long time ago, but there was a split a long time ago. So here's humans, and here's the rhesus monkey, and there was a common ancestor at one time, but there was a split, a diverging of our populations. And so we have some ancestors here that led to the rhesus monkeys that were different from ancestors that led to the human population. Now, we indicated that gorillas are more related to us, and so that means that there was a more recent common ancestor between us and gorillas. So we would put them right here. So when we see a phylogenetic tree and we have organisms right next to one another, this indicates that they're more related to one another. So let's indicate that we are more related to gorillas than we are compared to the rhesus monkey because they're further apart on this phylogenetic tree. Now let's take a look at an example AP question prompt. And there's a lot of information here. And when you read through this, the only information that is really important for you to answer this question, which is which of the following is the best explanation for this observation, is this statement right here. The fact that we find this sequence of this gene in all multicellular organism, it indicates a a common ancestor. So the fact that we find this gene in all of these multicellular organisms is an example of a molecular homology or a biochemical homology. Now, the second piece of evidence that indicates relatedness between species are called homologous structures. Now, I know I used the term homologous in the previous evidence that we discussed, and we talked about molecular homologies. Remember, homologies just mean similarities. When we're talking about homologous structures, we're talking about anatomical body structures that are similar between different species. And sometimes, instead of saying homologous structures, then we're going to say anatomical homologies. And it means the same thing. Remember, in science, we give everything 2 to 3 names to confuse you. So here's some examples of homologous structures or anatomical homologies. The flipper of a whale, the leg of a cat, the arm of a human. They're examples of homologous structures. Now let's take a look at those. This is the bone arrangement that we have in the forelimb of a human. And we can see here a lizard and a cat, and here's a whale. The fact that we have similar bone structures in different species indicates a common ancestor. When we take a look at the bones that are arranged in forelimbs, we can see the structure of bones here, which are called the carpals, and then we see the metacarpals, and then we see the phalanges. And the fact that they're arranged in a very similar manner in different species indicates there was a common ancestor. So here's what you're going to have to be able to do with homologous structures or anatomical homologies. First, you need to be able to list me an example of homologous structures that indicates relatedness. So if you use this example with the forelimb of humans, the leg of a cat, the flipper of a whale having similar bone structures, then you're giving me an example of homologous structure. But you also need to be able to use information to draw conclusions about relatedness. So here's an example where it says, use the information above to determine and justify the species that is more closely related to dolphins. Now, before we had this lesson, you might think that since sharks and dolphins have such a similar shape, that they must be more closely related. Well, their outside shape is very similar. But if you look at the anatomy of a shark and you compare that to anatomy of a dolphin, there's not a lot of similarities. So sharks and dolphins are related, but they're very distant relatives of one another. As a matter of fact, the dolphin is a closer relative to us than it is to a shark. When we take a look at their flippers and we compare them to the bone structures in our forelimbs, we see a lot of similarities. So we would be able to justify that the dolphin is more closely related to humans because they have this homologous structure. Their bone arrangements and their flippers are very similar to the bone arrangements in the forelimbs of humans. Now let's read through the next 2 example questions that I'm giving you. And what you need to be able to do is you have to determine what type of evidence would be most useful to determine relatedness between 2 different species. So I've given you some examples of evidence we use. Now you need to be able to determine what type of evidence would be the best at helping us make conclusions about the evolution of different species. So take a second to read through this problem and come up with the correct answer. Now, the reason why I do not like option A is because usually when we're trying to determine relatedness, we're looking at similarities between 2 species, and this is indicating that they have different niches. So I would say that's not good evidence that offers that they are related. The reason I don't like option B is because just looking at what modern species are doing, what behaviors they have, is not the best way to determine relatedness. C is your correct answer here, and we're using this idea of homologous structures to determine relatedness. The fact that we see the skulls of birds and crocodiles having this similarity here, this is a homologous structure. So similar structures in different species indicates a common ancestor. So the fact that they have this similar structure indicates relatedness. The reason why I don't like D is just because we can date them back to the same period of time doesn't necessarily mean that they're related. Now read through the next problem and see if you can determine the correct answer. So what you have to do in this question is you're basically having to determine what type of evidence would be most useful to determine relatedness. So we're trying to determine the relatedness between tetrapods, which are basically vertebrates that had limbs. So, for example, a frog is a tetrapod, and lobed fin fish. The reason I don't like A as an option is because you're just trying to gather data about lobed fin fish. This doesn't say anything about gathering data about tetrapods and then comparing your data or comparing your observations. The reason I don't like B is because if we are measuring the proportion of radioisotopes, all we're trying to do is we're trying to date the fossils. We're just trying to determine how old they are. And when we have that information, it doesn't necessarily give us information about how related they are. C is your correct answer. And once again, we're trying to use information from homologous structures to determine relatedness. So we're looking at the arrangement of bones in fins of the lobe fin fish and the limbs of the earliest tetrapods to see if there's any similarities, and that would indicate relatedness. Now let's talk about the information in this box first before I talk about why I do not like options D and E. Sometimes our best option to determine ancestry and relatedness is to look for homologous structures. And the reason why is because oftentimes these structures are preserved, but DNA gets destroyed over time. As a matter of fact, the oldest DNA that's been recovered is approximately only 500,000 years old. And scientists have estimated that even in the most ideal preservation conditions, that DNA would be completely broken down within 6.8 million years. And if you think about when the dinosaurs went extinct, they went extinct about 65 million years ago. So we're not able to obtain DNA from really old fossils and sequence that DNA. So that's one reason why I didn't like D, is because we're sequencing DNA from fossils, and yet we don't know how old they are. But you got to watch out for answers that say we're going to sequence DNA from fossils. But the fact that we're just trying to determine how big their genome is, how many base pairs long it is, that doesn't tell us anything about relatedness. If we're analyzing the DNA and we're trying to see similarities in base sequences, that would give us an idea of relatedness. But just how much DNA they have or the size of their genome is not going to tell us how related they are. And the reason why I do not like E is because it says we're looking for DNA in ancient fossils to determine the similarities in base sequences. And the fact that these fossils are ancient means that we're probably not going to get DNA from them to analyze. So in this situation, the most useful information would come from looking at homologous structures. The fourth type of evidence that gives us ideas about evolution of species are called vestigial structures. Vestigial structures are remnants of functional structures that we see in modern species, but they have no function in those modern species. So what this indicates is these structures were structures that were in an ancestor, and the ancestor utilized these structures for a certain purpose. But over time, we no longer use these structures, but we still have the genes for them that cause the development of these structures. So here's some examples of vestigial structures. We see wings on ostriches, but there's no use for the wings because ostriches don't fly. We have wings on penguins, but again, there's no use for them because they don't fly. So why do they have those structures? It's because they had an ancestor that did fly. Why do we see pelvic and leg bones develop in snakes and whales? Because obviously they don't walk. So again, you can see that there is no use of these structures in our modern day species, but they still develop them because they have the genes for them and a common ancestor walked. Some human vestigial structures include the tailbone. So we have the development of tail vertebrae inside. You can't see the tail on the outside, but we still have these structures that develop, yet we don't have external tails. We have ear muscles, even though we don't move our ears, and we have no need to move our ears like other organisms can. We have an appendix, and there still today is no known function of the appendix. There's some theories out there that it does serve a purpose, but it is commonly believed it really has no validated purpose. We don't know why we have an appendix, but maybe it was important in an ancestor. Homologies, or similarities in embryonic development. suggest ideas about the relatedness and the evolution of different species. So let's take a look at some embryonic homologies. One thing that we find in all developing embryo is the development of these pharyngeal pouches, and sometimes we call them gill slits. And you can see them labeled here on each of these diagrams. And we have a fish and a reptile, a bird, and a primate, a human. The fact that all organisms develop these embryonic homologies indicates a common ancestor. Now, they develop into different structures. Those gill slits are going to develop into gills of fish and amphibians, but they will develop into the ear cavities in primates. Another example of an embryonic homology that we see is what is noted as a post-anal tail. That just means a long tail. And we see that in all these developing embryos. So again, the similarity just indicates a common ancestor. Another piece of evidence that we look at are what we call transitional species. Transitional species give us an idea of the changes that took place between an ancestor and a modern species. So for example, we believe that horses evolved from a small four-legged creature that used to have 3 toes. And of course here we have modern horse, and it just has 1 toe. And if this is the case, then we should find transitional species that show the changes of this species over time. As a matter of fact, we do. We have found fossils that show 3 toes, and then we see that it looks like the 2 outer toes are getting smaller, and it's mostly just 1 toe that is being used. And so we see these changes over time. We see these changes in these transitional species. So they really give us a clear picture of how a population has changed over time. We have a lot of transitional species that show us the evolution of whales. We theorize that the whale species evolved from a land-dwelling mammal, and over time they became better situated to hunting in the water. If you think about it, they're mammals just like us. They don't have gills, they have lungs, they have to hold their breath to die for long periods of time. They have live birth, they produce milk, and they have hair on their bodies. And all those are characteristics of mammals. We have found transitional species that show how the hind legs have changed over time. And remember, there's still remnants of these leg bones. We have vestigial structures, the remnants of a pelvis, the remnants of femurs that we find in our modern day whales. So again, we're just using transitional species to show us how a species has changed over time. We theorize that birds evolved from reptiles. The reason why we believe that is because we have found transitional species that have characteristics of both birds and reptiles. One transitional species that's kind of famous is called the Archaeopteryx. And we have a well-preserved fossil of an Archaeopteryx. In this fossil, we can see the feathers. We can see their wings. We can tell that they have scales on their legs. Reptiles have scales. So we see a lot of similarities between reptiles and birds, and so that's why we believe there's a connection. That's why we believe that birds evolved from reptiles. Now, we don't believe reptiles evolved from birds because we can't find fossils of ancient birds. First we see reptiles on the fossil record, and then we see birds, or we find then fossils of birds, and they do not date back before reptiles. That's why we believe that birds evolved from reptiles and not vice versa. If we're going to use fossils to help us determine the evolution of species, then we have to be able to date them so we can draw accurate conclusions. Fossils are dated 2 ways, through absolute dating and relative dating. Absolute dating is used to determine a specific age of a fossil, or maybe even the rock around the fossil. So we can assume that if we date the rock back to 6 million years ago, that fossil was also around 6 million years ago. The result of absolute dating is going to be a specific age. So we're going to determine something like this fossil is approximately 3 million years old. When we're doing absolute dating, we are usually using the decay of radioactive isotopes to help us determine the age of the fossils. So, for example, what I mean by that is carbon-14 atoms will decay to carbon-12 whenever they lose neutrons. It takes approximately 5,730 years for half of the C14, carbon-14, to decay to carbon-12. And this is called the half-life of carbon-14. Now, other atoms that will decay, they have their own unique half-lives, but this is the half-life for carbon-14. Now, you might be asked to read a graph like this that's showing you half-lives and the amount of carbon-14 in a sample, and determine the age of the rock, or the age of the fossil. So it says, how old is the fossil that contains 50% of carbon as carbon-12 and 50% of the carbon as carbon-14? Well, we know that it takes 5,730 years for half of the carbon-14 to decay to carbon-12. So when we find a sample and the ratio is 1 to 1, 50% of carbon is carbon-14, 50% is carbon-12, and we know it took this many years to get there. So our answer would be 5,730 years. So how old is a fossil that's going to have about 88% of its carbon as carbon-12 and 12% of its carbon as carbon-14? Well, if we look at the graph, we can see that when about 12% of the carbon is carbon-14, then that means that we've covered 3 half-lives. So we would have to take 3 times the half-life of carbon-14, and that would give us the answer. So that would be the age of the fossil. Now let's take a look at relative dating. Relative dating cannot give us a specific age of a fossil. It can just give us an idea about the order of events. Basically, which organism was here first on Earth and then which ones came later. So basically, with relative dating, we're just simply looking at the layers of the rocks and knowing that the ones that will be further down were deposited first. So they're going to be older. And the layers on top are going to be the youngest layers that have been deposited. So we should be able to make a conclusion that these fossils down here are older than these fossils that we find here. So this species was on Earth before this species. So that's all we're doing when we're using relative dating techniques. Now, what you're going to have to be able to do is use relative and absolute dating methods to draw conclusions about the evolution of species. So go ahead and take a second to read through this problem and see if you can answer A. So this is actually an example of an AP release question. So I think this is a very easy question. It says that we're trying to find a link between lobed fin fish and amphibians. And if that's the case, then we should be able to find transitional species that would show us some of the changes that have come about as those lobed fin fish populations were evolving into these amphibian populations. So it states that we've dated fossils of the lobed fin fish back to about 380 million years old. So let's indicate that. And it says the oldest amphibian-like vertebrate animals that we can find are dating back to about 363. So they're not as old. So if you think about relative dating, then we're going to find the fossils of the amphibians in higher layers than we are going to find these fossils of these lobed fin fish. So the question says 3 samples of rocks were taken. So we have a sample of rock that's 350 million years old, 370, and 390. And it says select the most appropriate sample of rock in which to search for a transitional species. Well, again, transitional species happens as populations evolve. So we should find transitional species in the middle of when we're finding the lobed fin fish and those amphibians. So you should have selected that we need to look for them in samples of rock that are about 370 million years old. So basically, we selected the most appropriate sample of rock, and we just did a justification of our selection. And technically, we're just using ideas of relative dating to answer this question. Other evidence that supports these ideas of evolution is biogeographical. And basically, we're just studying the distributions of life in different areas of the world. Observations that biogeographers make support the ideas of evolution. We can believe 2 things about these butterflies that we find in different areas. We can believe that they were all made at the exact same time, or we can believe that they evolved and there was a common ancestor for all of them. Now let's think about if we thought that all of these varieties and species of butterflies, if they were all created at the exact same time, that no evolution occurred to create these different species. If that was the case, then why are they all located in South America? If they were all created at one time, then how come we don't find some in this location, some in Africa, some in North America, some in Australia? So the fact that they're all in one area indicates that there was a population that originated in that location, and some members of their population spread to nearby areas and colonized those areas. And those populations changed over time due to natural selection. Because when they move to a different environment, there's going to be different selective pressures. And so you're going to see different phenotypes arise in those populations, which are helping individuals survive better and reproduce. So because we find closely related species in a close geographical area, then it indicates that those populations of species got there due to evolution, and they weren't just made all at one time. The last piece of evidence that we're going to look at is the fact that we can actually observe the evolution of populations. So we do know that populations are changing over time. For example, here's some things that we have witnessed on the short time that we've been here on the planet. We've seen the development of strains of bacteria that are now antibiotic resistant. We see the rise of plant varieties that are resistant to herbicides. So you spray an herbicide to knock out the weeds in your crops. When you spray that herbicide next year, if there's any plants that are resistant, then your herbicide isn't going to work on that population of weeds that is now growing in your fields. So we've seen the evolution of these species and these populations. We've seen the rise of insect populations that are now insecticide resistant. And there's even a group of scientists that have actually observed the speciation of some finches, the development of a brand new species of finch, and we'll talk more about that later when we talk about the definition of species. So the fact that we can actually observe evolution of populations indicates that evolution is occurring. Now let's get into phylogenetic trees and cladograms. I've given you some examples of phylogenetic trees and cladograms here so that you understand there's lots of different looks to them. So this one kind of has more of like a Y branching pattern. And then this one has like a straight line here, and you have these branches all come off of it. And then you have more of a kind of a square branch here. And we have, on some of them, you can see that we have indicated homologous structures, like tetrapod limbs and jaws and stuff like that. So they look different. So sometimes you're going to be asked to draw a phylogenetic tree based on the evidence that they're giving you, or possibly a cladogram. Now, honestly, you never have to explain the difference between a cladogram and a phylogenetic tree. Honestly, if you Google cladogram, then a lot of things that are going to pop up are actually phylogenetic trees. If you Google phylogenetic trees, then you're going to see lots of images that are actually cladograms. So as for the difference in them, you're never going to have to explain the difference between them. It's just phylogenetic trees are more based on time, whereas cladograms are more based on structures, homologous structures that we see in different species. But do note that you're going to have to make either a cladogram or a phylogenetic tree based on evidence that we're collecting about different species. Also, I want to note, and I'm going to add this into your notes, that these are always changing. In science, whenever we uncover additional information, then we have to revise our theories. So cladograms and phylogenetic trees are not set in stone. They're always being manipulated and changed as we discover more and more evidence. Before you have to create your own phylogenetic trees and cladograms, let's just take a second to read through one that's already done. Whenever we have a branching of a line, like you see right here, so we have that branch point, then that indicates there was a common ancestor at that time between two or more different species. Now, more recent branching, like you see here, just means that the two species further down, they're going to be more related to one another because there was just a recent branching. So there was a common ancestor not that far back in time is what that's indicating. Now you're going to see on cladograms and sometimes phylogenetic trees, They're going to indicate structures that are homologous between the species that are on the cladogram. And again, homologous structures are just similar structures in different species. So the amnion is a homologous structure that we find in mammals, lizards and snakes, crocodiles, ostriches, hawks, and other birds. It is not a structure that you're going to find in amphibians. So the fact that these organisms share this homologous structure just indicates that they're more closely related to one another than they are to amphibians. Again, homologous structures indicate relatedness. Now, the organism that is kind of the odd one out, that really doesn't have any homologous structures that it's sharing with other species, they're referred to as the outgroup. So let's answer some of these questions about this cladogram. Are lizards more closely related to mammals or birds? Now, just because we have these two, lizards and mammals, right next to each other, it doesn't indicate that they're the most related. You got to take a look at the branching. So we have mammals, and here is our lizards, and here's birds. And you can see that when we go back, we see that there's a more recent common ancestor between lizards and birds, so our answer would be birds. Which species is considered the outgroup? So the odd one out that doesn't share homologous structures with other species is that outgroup, and that's going to be the lungfish. Identify the common ancestor to amphibians and crocodiles. So you just got to trace the line back to where it branches. So here's your crocodiles and here's your amphibians, and we can see that their most recent common ancestor was back at point 2. And then finally, identify evidence that suggests that crocodiles are more closely related to birds than to amphibians. So the fact that crocodiles and birds, they share this trait, or they both have the development of amnion and tetrapod limbs, indicates that they're more related than crocodiles are to amphibians, because the only thing that they have in common is tetrapod limbs. And the more similarities and the more homologous structures indicates greater relatedness. Now that we know what a phylogenetic tree or a cladogram looks like, then we're going to use some data to help us determine the relatedness and the evolution of different species. Here we've got a chart, and it's given us an idea of homologous structures. So what structures are common to different species? And whenever you see data given to you in a table like this, then I think it helps before you do a cladogram to rearrange your information. I like to indicate the characteristic that all possess or many possess up here at the top, and the one that is least possessed by organisms at the bottom. And then I also like to put organisms like the monkey and the rodent. I like to put them over here at the end because they are ones that have all of these different characteristics. So when I'm done rearranging my cladogram, then it looks like this. So once you see a table that looks like this, and there's like a stair step, I think it makes it very easy to draw a cladogram. So let's do it. We're going to go ahead and draw what I think would be the easiest one to construct. I'm going to show you that there's 2 different ways that you could draw this, and both are going to be correct. So what we're going to do to start off is we're going to draw a straight line. And the organisms that have all of these traits are going to end up here at the very end. So I go ahead and assign, and the end of this line is representing monkeys, and then here's rodents. Okay, so let's start back here with the starfish. The starfish only has one of these characteristics. It has eggs. So since it has less in common with the other ones, we're going to draw a line to branch off here, and we're going to indicate starfish. Now with the cladogram, we're going to have to indicate homologous structures, and the fact that all organisms have eggs, I'm going to put a line here. I'm going to put eggs, and anything basically downstream from this mark means those organisms have that trait. Now let's go to the next organism, frogs. So frogs have eggs and a backbone. So I'm going to take the frogs and I'm going to make a line for them. And I have to indicate that from here, everything down has a backbone. So frogs have a backbone, rodents have a backbone, and monkeys have a backbone. And then we look at birds. Birds have something that frogs don't. So we're going to indicate that here we get the evolution of warm-blooded organisms. So let's write warm-blooded. And let's label our line birds. And then we just have one more homologous structure to label, and it's homologous between rodents and monkey, and it's the fact that they have hair on their body. So this would be an example of a cladogram. Again, it's pretty simple once you rearrange the characteristics in the species in a table that shows a stair step. Otherwise, it can be a little tedious. I'm going to show you just a different way that we can draw this cladogram, because we can see cladograms that are shaped like this one, which is very similar to what we just drew. But we also have cladograms that are branched like this. So you just need to be able to make both of them and understand both of them. I will say on a previous AP test, you had to fill out a cladogram, and they already had one drawn for you. And it was more of this branched pattern. And I'm going to draw a branch pattern on this side. So here we have a common ancestor to all of them, but there is a split. So let's look over here, and the starfish is the biggest oddball. Doesn't share a lot of characteristics of the others. So I'm going to draw a line here, and I'm going to label it starfish. And I'm going to indicate here that all organisms from this point on have the ability to produce eggs. So that leads us to frogs, birds, rodents, and monkeys. Okay, so here is a common ancestor to them, and there is a branch. Now, we know that frogs only have two of these characteristics. They have eggs, and they also have backbones. So all these creatures from this point on have backbones. The frog doesn't have any other characteristics, so I'm going to go ahead and end its line and put a frog there. And now we have birds. Okay, birds, rodents, and monkeys. There's a common ancestor right here, but there was a split again. So I'm going to end the line there for a bird, and I'm going to write that all of these organisms are warm-blooded. I'm just going to put warm here. I'm kind of running out of room. And so we had another split, and then these organisms, there was a common ancestor between the rodent and the monkey, so we're going to show a split between those. And then we're going to indicate that they share hair on their body. And so we can either put monkey here or rodent there, doesn't matter. Let's go and put rodent, and then we would put monkey. So this is just a branch cladogram, and it's showing you the same information. You can determine who's more closely related and which species share a greater number of homologous structures. So let's do another example, and let me just show you how I would go about rearranging this table if I feel the need to do so before making a cladogram. So the table shows selected characters used to determine relationships among 4 species of mammals. Construct a cladogram to show the relationship between the species. If you want, you can just hit pause and see if you can construct a cladogram without any help, and you can check yourself. Now I just want to note that you could do this cladogram without rearranging any of the information in the table, but I'm going to just do so just to give you an example. I like to put the species along the top, and this one has a species along the top and the characteristics on the side. And what I'm going to look for is I'm going to look for a species that has all characteristics. It means that it's probably going to be the most advanced, and it's going to come at the end of the cladogram because advanced species do not evolve before simple species. So I'm going to put Neo over here, and I know that it's going to have 4 X's. And then the next one that has a majority of these characteristics is this one, Castoropsis, and I'm going to put 3 underneath it. And then you have the next one with 2 characteristics. I'm going to put 2, and then this one, and it only has 1. Now notice I'm also rearranging the order of these characteristics. So the characteristic that all 4 possess is here. So I'm going to put the tail, and the one that 3 have is 3 toes, incisors, and hair tufts. So now that I have a stair-step pattern here, then I can draw a cladogram pretty easy. So again, you're going to start out with a straight line, and the one that is most advanced is going to have all of these homologous structures is going to go at the end there. And then we have a branching here with this species, and this one, and then this one. And remember, the species that doesn't share a lot of characteristics of the other ones is going to be considered the outgroup. So just FYI, that one's considered the outgroup. If you see that term, that's what they're talking about. It just gives you an idea of characteristics of maybe more distantly related species, and you can compare those characteristics with these other three that are probably going to be more related. So let's add our homologous structures here. So we have all of them have a tail, and then we would put 3 toes here because 3 of them have 3 toes. And then only these 2 have incisors, so let's add incisors there. And then only this one has hair tufts. Now I just want to do one more for you, and I just wanted to show you how to do a cladogram, and you can see that we don't have a perfect stair step. As a matter of fact, you can see like gaps here, and then we will get another organism that had a segmented body and so on. So let's go ahead and set up a cladogram that's going to diagram all this information. So my most advanced organism here is going to be the human, obviously. So I'm going to go ahead and I'm going to draw my line. This will be a long line. So I'm going to draw a long line here. I know this last one is going to be human. We have a stair step, and we know that the sponge only has one of these different characteristics. So we're going to start out with the sponge, and we're going to put cells with flagella. I'm going to indicate that here. All species from this point down have cells that have flagella. And then we're going to add the jellyfish. And all organisms from that point have symmetry. And then I'm going to add the flatworm. And we're going to put bilateral symmetry. And then keep going with the earthworm. Just put this out further because I'm running out of room. Earthworm. And the earthworm and all these other ones have a mesoderm. So let's add mesoderm. Okay. And when we get here to the earthworm, we can see that it has this characteristic where its head develops first during embryonic development. And you see 3 other organisms have that. But the starfish and the human don't share that characteristic. So we cannot put head development here. So watch what we're going to do. Since that's a characteristic of just those 3 organisms, then I'm going to add a branch here and here. And right here is where I'm going to put head develops first. And so that means that's going to be a characteristic of our snail and our fruit fly. So you can see this one's looking a little bit different than some of the other ones that we've done, so that's why I wanted to show you this. And the idea that I have a human down here, if I put the head develops first here, then that's not a characteristic of humans. And so putting it on this line indicates that humans would not have that characteristic. I know that this characteristic is going to be found in these organisms that I haven't put on there yet. So I'm going to skip that. And I'm going to go down here. Since we have a line for earthworm, I'm going to go ahead and address this next characteristic, which is segmented body. Now, since that's a characteristic that is found in both the earthworm and the fruit fly, then it might indicate that they are more related to one another. So I'm probably going to change this a little bit. So you get to do a little bit of erasing, and that happens whenever you're making cladograms or phylogenetic trees. And I'm going to change this, and I'm going to put here is going to be the fruit fly. I'm just going to indicate fly. And then I'm going to go back and I'm going to put the snail here. And I'm going to show you what I'm going to do to show you that segmented body is a shared characteristic of earthworms and fruit flies. I'm going to put that right here. So I'm going to add for that little mark, that's going to be segmented body. And so that just means simply the earthworm and the fly show segments. Now, notice that the only organism that has a calcified shell is the snail. So to show that, I just simply go to the snail's line. And this is a trait that developed later on in the history of the evolution of this species. And it's just not a trait that you see in a lot of organisms. So they developed this because of the environment in which they lived through natural selection. So we're going to put calcified shell. Now you can also see that the fruit fly has a trait not found in other ones. The fact that it has a chitin exoskeleton. So let's indicate right here. I'm just going to add chitin here, since that's the polysaccharide that's found in their exoskeletons. And now let's go back to, let's finish up because we need the starfish and the humans. So let's go ahead and then draw a line over here for starfish. And then we already have our line there for human. We haven't marked these characteristics, and that is the fact that the anus develops first during embryonic development. That's why they're called deuterostomes instead of protostomes. These organisms that had a head developing first, those are called protostomes. Pro means before, so the front of them develops first. Only the starfish has a water vascular system, so we're going to indicate that. So we have a water vascular system. And then humans are the only ones that have vertebrae on this list. So we need to indicate vertebrae. So again, this one's just a little more complex, and you had some characteristics that were exclusive to certain species. So I wanted to show you how you show that on a cladogram. And also, just whenever you have to read a cladogram that is a little more difficult with more information on it, then you'll understand why we made these lines here. Because basically the only organism that has a water vascular system would be the starfish.